Hello everyone, this is Professor Santiago again. This time I'm going to be discussing about protein, functions, and its structures. Okay, so before we start, as always, let us first discuss about the objectives. Okay, so for objectives, first as we're going to be describing the main roles that protein play in organisms, we will be also describing the process in how proteins are assembled and also its structures. And we're going to be describing how amino acids differ from one another. Okay, so let's start. Now, 20% of the human body is made up of proteins, and proteins are usually large, complex molecules that are critical for normal functioning of cells. They are essential to the structure, function, and regulation of bodies, tissues, and organs. Now, let's discuss the amino acid structure and its classification. Now, every alpha amino acid has carbon atom, which is the central atom. Okay, this is the alpha carbon. Now, it's usually bonded with the carboxylic acid, which is the CaOH here. Then you have also the amino group or NH2. Then you have also the R group, which is very uh, unique for each amino acid. And then you have the hydrogen on one end. Further classification of amino acid is going to be based if it is a hydrophobic or a hydrophilic. Now, for hydrophobic, it's going to be based on the type of R groups there are. So if it's an alkyl or if it's an aromatic. Okay, so if it's an alkyl group, you have here glycine, alanine, valine, leucine, isoleucine, methionine, and proline. Then for aromatics, we have only two. We have phenylalanine and tryptophan. Now for the hydrophilic, when you say hydrophilic, this is water hating. Water loving, and this one is water hating. Now, hydrophilics are usually polar. Now, it's further classified into neutral, acidic, or basic. So, for neutral amino acids, we have tyrosine, serine, threonine, cysteine, glutamine, and asparagine. While for acidic, we have glutamic acid, aspartic acid, and for basic, we have lysine, histidine, and arginine. Now, let's classify amino acid based on its essentiality. So, we have what we call essential amino acids. Now, these amino acids are the ones that we need to take in as part of our diet because we cannot make them. So, an example of the amino acids we have histidine, isoleucine, leucine, methionine, phenylalanine, threonine, tryptophan, and valine. Now, for the non-essential, these are non-essential because we can actually synthesize this inside our body. Okay? And those are alanine, arginine, asparagine, aspartate, cysteine, glutamic acid, glycine, ornithine, proline, serine, and tyrosine. Now, amino acids, which is the building block of proteins, are actually bonded by what we call the peptide bonds. So they are linked by the amide groups, which we call the peptide bond. Okay. So if you will notice here, it's actually bonding between the amino group and the carbonyl group of the next amino acid. Okay. And water is removed in the process. Now, take note that in peptide bonds, the one that ends with the carboxylic acid group is called the carboxyl N or the C terminus or the CN. And the one that ends with the amino acid or the NH2 is called the amino end or the N terminus. Okay, so let's discuss now the proteins based on their structures. Okay. So the sequence of protein is determined by the DNA when it encodes during protein synthesis. Now, any change in the gene of the DNA sequence may lead to change in the amino acid sequence of the protein. Now, even just changing one amino acid in a protein sequence can affect the protein's overall structure and function. Now, to understand how protein gets its final shape or conformation, we need to understand the four levels of protein structure. So we have primary, secondary, tertiary, and quaternary. So let's first discuss the primary structure. Okay. So the primary structure is the simplest level of protein structure. So it's actually made up of amino acid sequence in a polypeptide chain. So we say polypeptide chain, you have a chain of amino acid linked with a peptide linkage. Okay. So here, if you look on the screen, you will see there the hormone insulin, which has made up of two polypeptide chains. So you have here chain A and B chain. Okay. Now you will notice that it is a sequence of amino acid and they are usually binded together by sulfur bonds. Okay. So you have here the amino acids in its sequence. Okay. So this is an example of a primary structure. Okay. So the next one is the secondary structure. Now, the secondary structure refers to the folding in between the polypeptide due to the interaction between atoms. Okay, so this is the most common type of structure is the alpha helix and the beta pleated sheets. Okay, now it's usually held together by hydrogen bonds between the carbonyl and the hydrogen of the amino group of another. Okay. So let's go into the specifics of the two types of secondary structures. So the first one is the alpha helix. Now, this is formed between the hydrogen bond between the fourth peptide bond in the carbonyl group and the hydrogen in the amino group. Okay. Now, it can either coil to the right or to the left, and it can also coil on other strands. Okay. So this is an example of the coiling structures for the hair. Okay, so this is your alpha helix. You have actually two helix forming or coiling on each other. Okay, now the second one is the beta sheets. Okay, now the beta sheets forms rigid structures with hydrogen bonds. Now there are two types of beta sheets. This is the anti-parallel. Okay, and the parallel. Now, when you say anti-parallel, it runs on the opposite direction of its neighbors. Remember earlier we discussed about C terminus or carbon terminus or carbon end and N terminus. So it means if this is the C terminus, the other one is the N terminus. So it's anti-parallel. While the other one is parallel. It means it's running in the same direction with longer looping sections between them. So it means if this is C terminus and if this is N terminus, all of this on the same layout is C terminus on this side, and then the other one is all on the N terminus. Okay, so that is for beta sheets. Okay, so let's go ahead and move on to the tertiary structure. Now, tertiary structure is important um, structure because of the hydrophobic interaction, which is wherein the amino acids with the nonpolar hydrophobic R groups cluster together inside the protein, leaving the hydrophilic amino acids on the outside to interact with surrounding water molecule. Okay, so you have here the hydrophobic structures. Okay, and then another thing is that it also have disulfide bonds. Okay, here are disulfide bonds which causes them to form bonds within itself. So usually we can find this between cysteine, okay, which is much stronger than the other types of bonds that are available. Okay. So aside from that, we have also hydrophobic interactions here. Okay. And, okay. Now the last one is the quaternary structures. Now, when you talk about quaternary structures, these are actually multiple peptide chain subunits come together to form the quaternary structures. Now, an example of the quaternary structure is hemoglobin. Now, it carries oxygen to the blood and it's actually made up of four globular units. Okay, you have two alpha globular units and two beta globular units. Now, usually when you talk about protein structures, it is important for them to retain its shape. Okay, so when a protein loses higher order structure, particular but not its sequence of amino acids, it's considered to be a denatured. Now, once the protein has been denatured, it loses its functionality. Okay? Now there are different reasons why amino acids or not amino acids or proteins loses its functionality. First one is heat. An example of this one is egg whites. When you cook egg whites, it loses its characteristics because it's already cooked. Okay. Next is another function or another cause of denaturation is mechanical stress. So when you beat egg whites, 'di ba? Pag nagbibit tayo ng egg whites, we tend to form icing, right? Lagyan niyo ng konting sugar, you'll have icing. Now, this icing is another kind of denaturation because the egg whites are no longer clear, transparent, and it becomes now a colloidal material. Okay, so you have heat, mechanical stress. Another one is strong acids. So you have acids when you cook fish. Remember yung kinilaw or kilawin, di ba yung fish, lalagyan nyo ng suka or calamansi, it will cook the meat. That is denaturation. Okay? So those are some of the causes of denaturation in proteins. Okay? Now let's classify proteins based on biological function. So the first one is that proteins also functions as enzymes. They actually form part of catalytic activity. They also regulate biological reactions inside the cell. Another function of proteins is transport. Okay, so these are proteins that helps in maintaining life by transporting nutrients and gases. So an example of that one is hemoglobin in the red blood cells. Okay, and then you have also blood plasma that contains lipoproteins that carries lipids from lipid to other organs. Okay. Another function is storage protein. So these are actually stored inside the cell of the tissues that functions as to serve food and can be utilized over time when it is needed to provide energy. So an example of that one is casein, which is storing proteins in milk, and ferritin that stores iron in spleen and liver. Next one is for contractile or motile movements. So an example of that one is the muscles. So you will notice that you have there proteins, actin and myosin. Okay. So you have the myosin here and then you have here moving inside the muscle. Okay. So you have the actin and myosin which is present in filamentous protein in muscle cells for functioning in contraction of the muscles. Next is structural protein. So an example of that one is the tendons, cartilages, bones. Now, you also have collagen, which is a fibrous protein. Then you have also ligaments, which contains elastin. Okay, that is responsible for stretching it into two dimension. Now, keratin can be found in hair, fingernails, yung feathers ng birds, that's your keratin. And then you have also the protein fibrin, which is a component of silk fibers and threads of spider web. Another function is hormones. So some proteins help to regulate cellular and physiological activity. Okay, so you have there the pituitary gland. Then you have also an example of that one is insulin, growth hormone, parathyroid hormones, and also repressors. So that is for biological function of proteins. So the next classification is based on shape and solubility. So the first one is fibrous protein. So these are proteins that have a rod-like structure. They are not soluble in water. Now they are held up by polypeptide chain that are parallel to the axis and are held together by hydrogen and disulfide bonds. Now they can be stretched and contracted like thread. Now examples of this one, you have collagen, keratin, fibrinogen, muscle protein, just to name a few. Now, the second one is globular proteins. Now, these proteins more or less are spherical in nature. Now, this is due to their disruption of the amino acid. So, they are hydrophobic inside and hydrophilic outside, and making them very soluble in aqueous solution. So, examples of this one is myoglobin, albumin, globulin, casein, and hemoglobin, and other enzymes and protein hormones. Okay. Another classification of proteins is if it is a simple. Okay. Now, when you say simple, it contains amino acids which are doesn't have major organic hydrolysis products. So an example of that one is egg, serum, which contains globulin, wheat contains glutenin, and corizinin, which is coming from rice. Now, on the other hand, you have now conjugated proteins. Now, we say conjugated proteins. These are uh, proteins that only give amino acid but also contain other organic and inorganic components. So, it may contain non-amino acid as part of this prosthetic group. Okay, so usually these are classified uh, for proteins based on the presence of their prosthetic group. So an example of that one is lipoprotein. So the prosthetic group is lipid. So this is an example is beta lipoprotein of blood. Glycoprotein, which is containing carbohydrates. An example is immunoglobulin G. Phosphorate, phosphoprotein contains phosphate group, which is found in casein of milk. Hemoprotein, which is heme or iron porphyrin, which is an example protein is hemoglobin. Flavoprotein, you have flavonucleotides, and example of this is succinic dehydrogenase. Metalloprotein, which contains prosthetic groups of elements like iron, zinc, calcium proper, and molybdenum. And the example protein is ferritin, alcohol dehydrogenase, calmodulin, plastocyanin, dinitrogenase, and then nucleoprotein. An example of that one is RNA, and an example of that for the protein is the ribosome. Okay, so that is for simple versus conjugated proteins. Now, the next one is based on its nutritional value. Now, its proteins can either be classified as a complete protein, where it contains an adequate amount of all of the essential amino acids that should be incorporated in a diet. So an example of that one is fish, meat, poultry, cheese, egg, yogurt, and milk. Okay. So it contains, so when you say complete protein, it contains all the amino acids essential to build new proteins. Okay. So it does not lack, it contains both essential and the non-essential amino acid. Okay. Now, incomplete proteins, on the other hand, okay, so it is lacking some essential amino acids, even in correct proportion, and that's why it's called as a partial protein. So even if it contains some essential, but if it's lacking the others, it's still considered as an incomplete protein. So an example of those are grains, nuts, beans, seeds, peas, and corn. Okay? So those, that is the difference between complete protein and incomplete protein. Now, since we have issues about complete and incomplete protein, so what normally happens is that we need to combine foods that contain two or more incomplete protein in order for a complete protein to be created. Okay, so if in case there are amino acids that are missing from one food, you compensate it with adding another protein that contains that missing amino acid. Okay, so an example of that one is grains with legumes. So you have lentils and rice with yellow peppers. Nuts with legumes, so you have their black bean and peanut salad. Now, grains with dairy, you have cheddar cheese. So you have cheddar cheese and whole wheat pasta. Then you have dairy with seeds. So you have the yogurt mixed with sesame and flax seeds. Then legumes with seeds. So spinach salad with sesame seeds and almond salad dressing. So medyo social, yes. Marami kang kakainin, but one thing to remember is that it's gonna fill up or complete the missing amino acids in your diet. Okay? So that is all for classifying proteins. So let's go now to detection of amino acids. Okay. So the first test that we use in detecting amino acid is the Burette test. So usually it identifies the presence of the peptide bond. Okay. So when treated with copper sulfate solution in the presence of alkali, sodium hydroxide, or potassium hydroxide, protein reacts with the copper ions to form violet colored complex called burette. So here is the negative result for burette test, and here is the positive result for burette test. Okay. The next one is xanthoproteic test. Okay, so it is an identification test for proteins, and it gives a positive result for those that carries aromatic group. Okay, so when protein is treated with hot concentrated nitric acid, it gives a yellow to orange colored substance, this form. Okay, now it usually gives a positive result for proteins that contains tyrosine and tryptophan. So here, even after heating, if that protein does not contain tyrosine and tryptophan, you're just gonna have a faint yellow color, but it's not going to form this kind of coagulation, okay, or substance. Next step is the Nene-Hydrin test. Now, this is a test for amino acids and proteins with free NH2 group. Now, the NH2 group reacts with Nene-Hydrin to produce the blue colored complex. So here on this diagram, it shows there if there are no amino acids, it's just going to be clear. If it is showing a purple colored complex, it means amino acid is present, and this is the positive indication of amino acid. But there is one exemption to the rule. Proline gives a yellow result for ninhydrin. This is the only amino acid that gives a yellow color result for ninhydrin, but all of them are usually around the purple violet colored complex. Okay, so that's for ninhydrin. Now the next one is the Millon's test. Now the Millon's test is used to identify tyrosine inside a protein. Okay, so it's specific for tyrosine. Now, when the Millon's reagent is added to the protein solution, it gives first a white colored precipitate, and then when you boil it, it will be turning to brick red. Okay, now that is how it's gonna look like. So this one has been heated, and this is the upper part wherein the mixture was not heated, okay, for visualization purposes. Now, take note that gelatin will not give a positive result because it does not contain tyrosine. Okay. Now, next one that we use to test for proteins is chromatography. Okay, so chromatography actually means color, or from the word chroma, and graphene means to write. So it's actually meaning color writing. Okay, now it was actually employed by scientist Mikhail Chevet to identify and separate the different color substances in plants. Now, in chromatography, it's actually composed of two parts: the stationary phase. Okay, so it can be solid or liquid. And then you have now the mobile phase, okay, which is moving over the stationary phase. So most of the time it is liquid in nature. So when the mobile phase is placed over the mixture of the stationary phase, the components of the mixture will gradually separate from one another. Okay, so that is for chromatography. Now we have two types of chromatography available. So the first one is the adsorption chromatography. Now under the adsorption chromatography, we have column chromatography. So this one is the stationary phase, okay, inside a column, okay, a chromatography column. Now, what would happen is that you're going to be placing the mixture that's supposed to be separated. Then you will constantly add the mobile phase in the reaction. So if you will notice, at the first, on the initial step, when you added the mobile phase, you have here the components starting already to separate. Now, as you gradually add more mobile phase into the solution, it will tend to separate. So usually the heavier or the amino acids with a heavier molecular mass will tend to flow out or be pulled down by gravity as the mobile phase is constantly added, followed by the average amino acid with the average molecular mass, and then the lightest will be the last one to go down the column. Okay, so this is the concept for column chromatography. And then in the end, you'll just be collecting it as it passes through the column. Okay. So that is for column chromatography. Now the next one, this is called thin layer chromatography. Now it's also in adsorption chromatography, wherein it's going to be using a thin layer of absorbent coated on a glass plate. Okay, so the stationary phase is usually a silica or gel or alumina on a glass plate, and then you will be placing a spot of the sample inside, and then you have there a solvent system. Okay. Now, what would happen is when you place it inside, due to capillary action, the solvent would rise and it will carry the sample upwards. Okay. Now, usually over, you're going to be using this after a certain, or allow it to climb upward into the solvent or into the stationary phase, let's say an hour. And afterwards, you're going to see that you'll have there the multiple um, components of the sample separating. Okay, again, the lighter ones are being carried forward faster. The heaviers are going to be carried last. Okay, now afterwards, after the 1 hour session has been completed, you will be identifying the distance traveled from the solvent and the distance traveled by the solute. Okay, so this is your TLC plate or thin layer chromatography plate. You have your sample. Now, this is the line of your sample. Okay, and this is also the line of your solvent. So when you measure this after 1 hour, the distance from this point or the baseline to the solvent front, this is actually the area where the solvent traveled to. Okay, you're going to be identifying this, and then you're going to measure the distance from the baseline and the distance covered by each component that was separated on the TLC plate. Now, you will be using the retardation factor formula. Now, the retardation factor formula is actually using the formula RF or retention factor, distance traveled by the component from the original line, and over the distance traveled by the solvent from the original line. Okay, so you use a calculator, of course, to use to calculate for this. Joking. Okay, now once you have already, now what if you are not able to see the samples? Okay, now we usually have ways to detect the components on a TLC plate. Now, if it's color spot, you can see it right away via eye. Kitang-kita. Then the second one, if it is colorless, but it usually glows under ultraviolet light, use an ultraviolet light. Then it you can also use a cover jar containing iodine crystals. So what would happen is that the components of the sample will absorb iodine, producing now brown spots. And you could also use ninhydrin. Okay, so it will give a purple complex color upon spraying it on the thin layer chromatography, and then you'll be able to identify the position of your samples. Okay, so that is for adsorption chromatography. Now, the last one is partition chromatography. Now, the difference between the partition chromatography and the adsorption chromatography is that for adsorption, the stationary phase is solid, the mobile phase is liquid, while for the partition chromatography, both components are made up of liquid. Okay, so your stationary phase is liquid, your mobile phase is also liquid. Okay, so in paper chromatography, which is an example of partition chromatography, so you will have there a paper, a special paper or chromatography paper. Now you will be placing a sample on the original line. Okay. Now, afterwards, you're going to place it inside the developing chamber. Now, inside the developing chamber, you will have there the solvent. So it's a usually mixture of ethyl alcohol, butanic acid, and water. Okay. And then Make sure that it's not bent and cover it with lid. Now, let the solution or the solvent to go up into the paper, chromatography paper. Okay, so after 1 hour, you go back to the sample and you will notice that your solvent line has already reached almost the top. And then you will notice some of the components of these samples are separated into its base components. Okay. Now, it's almost similar with the TLC or thin layer chromatography. You're just going to be measuring the distance of the solvent line from the baseline for the denominator of the RF. Then each sample units will be having, you're going to take note also of the distance traveled by each sample compared with the baseline. Okay. So yeah, so it's going to be using also the RF, the retardation factor or the retention factor. Okay, so that is for paper chromatography. Now, you will be proceeding with the different laboratory activities, and your professor will be assisting you forward. Thank you for listening. Have a nice and lovely day. Bye.